Hello, class. So this is our new series on protein turnover and amino acid catabolism. This actual topic will be broken down into different subcategories. So for this particular part of the series, we'll be focusing on proteins are degraded to amino acids and protein turnover is tightly regulated. So the proteins that we get from our diet is vital for a source of amino acids within our body, especially the essential amino acids that we cannot actually make synthesize in our own body. We have a list of essential amino acids that are found right here, so you should start to become really familiar with these particular amino acids because these will be coming up further within the course and with testing questions. And so proteins ingested in the diets are broken down into amino acids or small peptides, which will be then absorbed by the intestine and then transported in the bloodstream. So this is another crucial source of amino acids by having the proteins degraded down into amino acids. So how is this process happening? So you basically have proteins and that get broken down by proteolytic enzymes. Then these enzymes are living within your stomach. Proteolytic enzyme is also called pepsin. They can function in highly acidic environments, and so they start breaking down your proteins into amino acids and oligopeptides. And so before it actually goes into the intestinal cell, it also needs to be neutralized. And so the pancreas has sodium bicarbonate, which neutralizes the pH of the food, and a variety of pancreatic proteolytic enzymes that is breaking down the proteins within our bodies that we ingested by our diets. And so within that, we have peptidase that is located within the membrane of the intestinal cell. And so what they do is to break the peptides, um, illegal peptides, like break them down even further. It goes from the amino terminal end. And so this note that there are, even though this is showing three, there are at least 7 different transport that exist. Each specific each that is specific to a different group of amino acids to help transport the amino acids into the intestinal cells. And so once it is transported into the intestinal cells, you have amino acids, but you also, the legal peptides were broken down further down to tripeptides and dipeptides. And there are more peptidases to break them down into amino acids that can now be transported into the bloodstream via sodium amino acid antiporters. And so when you have these proteins that been ingested, you actually have something called ubiquitin, which serves as a protein that targets other proteins to be destructed and be broken down. And so what happens is there is a terminus C that is glycine residue of the ubiquitin, and that is what attaches onto the lysine of a protein residue to produce a isopeptide bond, and there now this protein has been targeted to be go through the process of degradation. And so the ubiquitin also has some lysine residues. It has them, so other ubiquitin can actually attach to these lysine residues as well to make it even more of a target protein to be degraded. And so this formation of this isopeptide is done by ATP hydrolysis. And so for this ubiquitin to actually, this is the ubiquitin and have that carbon terminus to eventually go on to the target protein for it to be targeted for degradation. This involves 3 enzymes, so it's very important to remember this is 3 enzymes that are being involved in this process. So first, utilizing ATP, the carbon, remember this is from the glycine residue carbon source that bonds to the AMP, and then from there it can be transferred on to the first enzyme 1, adenylate ubiquitin. And so you have that attachment to that enzyme 1, and then there is another transfer for that ubiquitin goes on to enzyme 1 to enzyme 2, and that is the cytosine residues of enzyme 2 that carbon terminus, which is the glycine residue of the ubiquitin, is attached to that enzyme 2, and then from there you have enzyme 3 that actually works as a way of transferring the ubiquitin on to the targeted protein, and then once that is transferred over, you have now the free E2 enzyme 1, enzyme 2, and enzyme 3 to be ready to use other ubiquitins that has been attached to the AMP to start the process all over again and target more proteins that needs to be targeted for degradation. And so for some sequence of amino acids that's in the protein, they have a specific sequence that's termed degron, and so that indicates that that protein should be degraded. And so this dependency is referred to as end rule or in degron. And so within that, sometimes when you have these specific sequence of amino acids that has a code to the body to say, hey, I need to be degraded, sometimes those signals is actually blocked by other sequenced amino acids, and so they just need to be exposed. In order to do that, there is some proteolytic cleavage that happens, and then now the degron is exposed, and now the body sees the sequence for that particular protein to be degraded. And so the amino acids that are in front that's blocking this degron signal is called pre-end degrons. And once those are released, then it exposed the signal for the protein to be degraded. And so since we have all of these different amino acids that are around, you can have highly stable residues, which means highly stable amino acid groups that makes it actually hard, it's a more stabilized type of sequence. And so these amino acids would degrade, cause that protein to degrade slower, and so it has a higher half-life. But when you have more so of amino acids that are more destabilizing, especially remember the lysine, what I mentioned previously was one of those target amino acids. Those are more destabilized and they'll break down a lot quicker. So be familiar with this list of amino acids that destabilizes as well. And then there are some amino acids will become destabilized residues after some chemical modifications. And so once these ubiquitin is attached to the protein, it's like, hey, I'm ready to be broken down. What happens next is it goes to the enzyme proteasome. And so then the protein is broken down to peptide fragments. And then what is spared is the ubiquitin to be released to target other proteins again. And so it gets recycled basically. And so once you have these broken down peptide fragments, then you have this enzyme proteolysis to break it down to free amino acids. And those free amino acids could either be left to actually do biosynthesis of proteins. We'll talk about that a little bit later. And then the free amino acids could also be broken down into just amino groups or the carbon skeletons. But if you just have the amino groups, they will go through a nitrogen disposal by urea cycle. So we'll talk more about the urea cycle later on. And then as far as the carbon skeletons, they can be used to create fatty acids or used as a fuel source for cellular respiration or used as a material for synthesis of glucose or glycogen. And so with these different proteins, there are a number of physiological processes are controlled at least in part by protein degradation through the ubiquitin and proteasome pathways. In each case, proteins being degraded are regulatory type of proteins. And what it helps out is these processes right here, those regulatory proteins. So kind of get familiar with this list as well. So for instance, it said gene transcription, cell cycle progression, even tumor suppression. These regulatory proteins kind of help out with these type of biological functions. And with that, that is the end of this part of the series, and we'll continue on the next part in the next lecture.